In this video, I'll be discussing the importance of what we call microRNA, or miRNA, and the role that miRNA plays in gene expression and consequently in cancer. To start, we first must understand what gene expression is. Recall the central dogma of molecular biology. A gene contains the genetic code to be transcribed into an RNA molecule, which is then translated into a protein, which carries out some function in the cell. Ultimately, all of the proteins with all of their diverse functions work together, allowing the cell to serve some purpose in the body. However, obviously not all of our cells are functioning the same way. This is because each cell has a specific set of genes that it expresses at any given time, and those genes are not all expressed in the same amount. Allowing differential expression of the about 20,000 protein-coding genes is what allows a neuron in your brain to look and act a lot different than, say, a muscle cell in your bicep. While it's essential to have differential gene expression in the cells of our body, it becomes problematic when the genes that are expressed at incorrect levels are involved in cellular processes like division, survival, or migration, processes for which dysregulation can cause a normal cell to become cancerous. Genes that promote such tumor formation are called oncogenes, while genes that suppress tumor formation are called tumor suppressors. So we don't want oncogenes to be expressed too highly, and we don't want tumor suppressor genes to be expressed too low. But how can this change come about? There are many mechanisms involved in regulating gene expression that can act at all steps in the pathway from DNA to protein. But this video will focus on just one: microRNA-mediated mRNA regulation, an essential way that gene expression levels are controlled and an important component of human health. So what is microRNA? Let's take it back to our central dogma. Generally, we understand that a gene is transcribed into mRNA is translated to protein, which carries out some function. However, this is not always the case. It turns out that there are many different types of RNAs that don't translate into proteins, but instead carry out their function as nucleic acids. Including in these non-coding RNAs are microRNAs. Often found in clusters, microRNAs are transcribed from the DNA and quickly cleaved by a protein complex called trosha into pre-microRNA. This pre-microRNA is then exported out of the nucleus and into the cytoplasm, where the enzyme dicer cleaves it into a mature microRNA of about 22 nucleotides long. Dicer actions initiate the microRNA's assembly with the RNA-induced silencing complex RISC. Within this complex, one strand of the RNA is removed, and the mature microRNA is ready for action. The effect of the complex on the target mRNA depends on the sequence homology that the microRNA has for its mRNA target. If the microRNA is very similar to the targeted mRNA, mRNA will be cleaved by the RISC complex. However, if the sequence has limited base pairing with the target mRNA, the complex will function to repress the translation of the mRNA. In either case, the microRNA RISC complex serves to lessen the amount of the targeted mRNA that is translated into protein, and thus lowers the expression of the gene that the mRNA transcribed. Due to its non-exact binding, it's been found that a single microRNA can target up to hundreds of different mRNAs, and thus can control the expression of hundreds of different genes. Considering that researchers have identified over 940 microRNAs coded in our DNA, it's easy to see how this regulation process could largely impact a cell if it went awry. MicroRNA dysregulation in cancer were first connected in 2002 when it was found that microRNA 15 and 16 were both deleted or downregulated in the majority of B-cell chronic lymphocytic leukemia. Normally, microRNA 15 and 16 target the pro-survival gene BCL2. Without their activity, BCL2 is more highly expressed and able to carry out its role of preventing a damaged cell from dying when it really should. Since this finding, discovery of dysfunction and dysregulation in the microRNA pathway in cancer has exploded. Other examples include microRNAs targeting a protein that inhibits a replication inhibitor, and those that target a protein that inhibits the well-known tumor suppressor p53. But these examples are only the tip of the iceberg when it comes to how microRNAs are involved in cancer. Our knowledge about microRNA offers a unique opportunity for studying and treating cancer. Researchers see the potential for microRNAs to serve as diagnostic biomarkers, therapeutic tools, and therapeutic targets. Theoretically, if we could deliver microRNAs to those cells that have lost them, or to target oncogenes that have become overexpressed in cancer, we may be able to exploit our endogenous system of gene regulation to our advantage. Limitations do, of course, exist. Delivering microRNAs in the correct cells to the correct targets in the correct amount remains a challenge. Despite these challenges, further research will continue to elucidate the role of microRNAs in cancer towards better, more personalized treatment.